All right, so McGill Scientific Writing Initiative presents our first workshop on reading research articles. It is Thursday, September 24th, 6 p.m., and we are very glad to introduce our first guest speaker. So please go ahead.
Okay, hey guys, thanks for having me, and I'm glad I'm able to present the workshop. Hold on, I'm just playing around with the screen a little. Okay. Yeah, so let's get started. So here's a little bit about me. So my name's Tommy. I graduated with a Bachelor's of Science from McGill in winter 2020, majoring in neuroscience. And I'm now a master's student, still at McGill, in the integrated program in neuroscience. So I do my research in Dr. Gabriela Gopi's lab at the Neurobiological Psychiatry Unit of McGill. Hold on. Okay. So I've been involved in scientific research since about 2018, where I worked as a summer research student in the orthopedic oncology department at the Montreal General Hospital. Over there, I was part of a team looking at the follow-up care of patients who underwent different types of treatment for soft tissue cancers. In 2019, I began undergraduate research at my current lab, and I stayed on as a summer research student and then as a student enrolled in NSCI 420, which is an independent research class. If it interests you, we can talk a little bit more about independent research classes at the end of my presentation. But very quickly, I encourage anyone looking to get involved in research to enroll in one of these classes because it allows you to get university credits for your research as opposed to working at a lab on top of the 5 classes or so that you're already taking. So my undergraduate research very quickly focused on characterizing the pro-social effects of LSD in mice. And now my master's project looks at how this can be applied to mental illnesses with antisocial phenotypes. Yeah, so maybe we could discuss research or anything like that later on. So here's a brief outline of what I'm going to present today. I'll start by quickly talking about the different resources that let us access scientific literature. Then I'll talk about the 2 main types of scientific papers. I'll break down the different sections of scientific papers. And I'll finally give you all some tips on effectively reading a paper and how to research a topic. There'll be time at the end for questions, but like Sydney said, feel free to unmute yourself and ask a question or type in the chat and maybe Sydney can ask. Yeah, so let's get it going. So the first resource I'd like to talk about is Pubmed. This website is a collection of essentially all scientific papers published in a major peer-reviewed journal. So the way I've organized my slides is that each screenshot of a web page is 1 click away from the next. So here we see the main website, and I've typed in CBD in the search bar. And so just by going to the next slide, it's as if we were on Pubmed. I think it's a bit easier like this. So after pressing search, we get here, and we can see the papers that best correspond with our search. We could have also included authors in the search or anything that helps us specify what you're looking for. After clicking on a paper you'd like to read, you get here. So this is an old paper that several of my colleagues wrote, and we'll use this paper as a model today because it's organized in a very classic way. Sometimes when you get here, all that you can see is the abstract, and I'll get into what the abstract is a little later, but essentially the problem now is that you don't have access to the full paper. From here, what we're looking for is a link to the full text, which I put in a red circle you can see to the right. It's important to note that clicking on the link above the one I've just circled, I'll go over with my mouse, it would ask for us for either credentials or for some kind of a payment to subscribe to the journal in which the paper was published. The reason the one that I've circled would give us the full article is because it says free. It says it right over there. And sometimes there's no link for a free full article, but I'll explain a very easy way to get the full paper in a couple of slides. Finally, you click that and you end up here, and you can read the entire paper or even save it as a PDF and read it later by pressing, here's a little PDF at the top right. So I prefer using Pubmed because it's how I learned to look up papers, but Google Scholar works just as well. Again, all I've done here is typed in CBD into the Google Scholar search bar, and it's compiled a list of papers. And once you click on one of these links, you'll end up at the same place that Pubmed brought you. So now if you do find a paper, which there's no free access to the full article, a very simple way to bypass this is to use the McGill Library webpage. So when you get to the main website, you press Advanced Search. And from here, you simply type in whatever keywords you're looking for in your search. This website then brings you to a list of papers that match your search. For example, I like to read the first one. So I click on it. And then I have access to several links for the full paper. And sometimes some of the links are broken, but in my experience, there's always at least one that works. Yeah, so I used to not think the McGill Library was too useful, at least the website, but when you're looking for full papers like this, it comes in pretty clutch, if I must say so myself. And then after that, you'll have to put in your McGill credentials. Finally, you end up with a full paper. And if your computer is connected to the McGill Wi-Fi, it'll automatically give you access to the full article directly from PubMed or Google Scholar. But if you're at home and you still need to access these things, you can do it like this. And here's a little tip I thought I'd include. So when you need to cite papers, you can often find the citations of the paper directly on the website with a full article, but sometimes it's not there. So in that case, again, using the McGill Library webpage, there's a little place you can click that says cite. And after you do that, you get the citation in whichever format you'd like. So here it is in APA. Now I'd like to talk a little bit about the different types of scientific papers. So in general, there are two main types of papers: original research articles and literature reviews. Original research articles are most often the ones that your professors will assign you. The authors of these papers conducted an experiment and are presenting the results and its implications. A literature review, on the other hand, does not present any new results to the scientific community. The authors of a review are simply gathering and condensing the existing literature of a topic and are presenting an overview of what they find and where they think this field is heading. So I've just included a screenshot of one. There's a relatively rigid format for research articles. However, there are some sections that are always found in original research papers and that are fundamentally different than the same sections found in literature reviews. For example, the methods section of an article will present the ways that the authors performed the experiment. I'll get more into this a little later on. And this contrasts the methods section of a review, which simply explains the way that the authors filtered the papers whose results they present. So you won't always find a methods section in a review, especially in a review in a relatively new field of research, because there aren't that many papers that need to be filtered through. Similarly,
I have a question for that.
Yeah.
Sorry, just to kind of like, because I'm not sure everybody's been exposed to this before. Would every single paper have a section like that, like a methods and then like a results section?
Absolutely. So I'm going to get really into the different sections a little later on, but reviews won't necessarily have these, like this structure, but articles, for the most part, do. I would say 99% of any scientific article you'll find will have these things.
Perfect. All right, thanks.
No problem. Yeah, so the results section of a research article will present the findings of the experiments performed. Conversely, you won't find a results section in a review because they have no results to present. And finally, research articles almost always have a distinct discussion section where they rationalize their results in the context of what is already known. On the other hand, literature reviews are pretty much one big discussion, so you won't usually find a dedicated section in them. So I've quickly mentioned some of the different parts of a scientific paper. Now I will go in depth about the information you'll find in each of them. I should mention that the rest of the presentation will mainly focus on original research articles. And the reason for this is that reviews are designed to be very easy to understand and their formats are not very rigid. Original papers, on the other hand, dive deep into the intricacies of their experiments and are usually way more difficult to understand for someone who is not an expert in that field. So the first section is the abstract. The abstract is the first thing you read when you look up a paper, and it is designed to give you a quick summary of the project. It briefly goes over the purpose of the study, the design, the notable findings, and the very quick conclusion of the results. It is meant to be an easy way for you to determine if the content of the paper is of interest to you. So abstracts are especially useful when you're trying to compile papers that have a common theme or a common finding. Here's an example. So if for some reason I'm writing a paper on the effects of childhood consumption of chocolate and obesity, I'd probably start the paper off by saying something like, chocolate is a snack that children have adored for decades, and the study aims to see if eating chocolate as a child leads to obesity in adulthood. So this is fine, but my paper would be a lot stronger if I reference a couple of studies that show that children do in fact eat a lot of chocolate, and that this is a study worth pursuing. And if there are tons of papers published about obesity and about chocolate, I need a quick way to sort through the ones that help prove my point, that children do in fact eat chocolate. So I would accomplish this by reading the titles and short abstracts of the papers I come across, and only reading the full article of the abstracts that seem like they'll have what I need. It's a lot quicker than reading every paper I come across from beginning to end. So chocolate and obesity, might not be the best example since it's something that most people assume to be true, but using other papers to back up assumptions you make are especially useful when the assumptions you're making aren't trivial. So here's the abstract of the paper I mentioned before and the one that we'll be using today as an example. You can see it's quite short and it probably only takes a minute or two to read. And now what I've done is I've erased some of the abstracts, so I show you how it's organized. So in this first couple of sentences, the authors declare the rationale of their project. They are saying that CBD, which is known to interact with the specific serotonin receptor, may have anti-pain and anti-anxiety effects. They also say that the effects of serotonin signaling and on neuropathic pain are not known. So it might seem like a lot of like scientific gibberish, but we're gonna go slowly to try to break down what each of these things mean. The thing that's important to understand here is that the authors are really trying to just set the stage for the study. The next part is the bulk of the abstract, and it simultaneously describes the way that the authors tested their hypotheses and their results. So it may look confusing, but it has many different acronyms and things in brackets, but if we read it carefully, it becomes much, much clearer. For example, here I've highlighted some of the key pieces of information that are fundamental to the study. So in red, we see some of the methods. So you see things like in vivo, single unit, extracellular recordings. So all this is, it's a technique that people use to measure brain activity. We also see rats. So we know that the authors performed this study in rats. We know that the authors inject intravenously and that they give CBD. And we see that the dose is right here, 0.1 to 1 milligram per kilogram. In blue, they mention the results of the experiment using the methods they just described to us. So it might seem like a lot, but bear with me. So we learned that CBD injected intravenously in rats at the doses they mentioned decreased the firing of serotonin neurons in a brain region called the dorsiraphe nucleus. The reason I'm trying to break it down like this is because, like I said, it might seem like all these terms that we might not understand, but when you break it down, the authors are really trying to get their point across as simply and as structured as possible. So they go on to explain some of their other findings, but the thing that's important to understand here is the authors are attempting to communicate to us how they performed their study and what they found in the most efficient way possible. So after you've read the abstract, you can decide if these results are interesting and if, to you, and if you want to read the full paper. I should mention that once you've read many papers of the same field, reading the abstracts becomes way easier because the acronyms, the drugs, receptors, techniques, they all become familiar and you won't need to Google them after coming across them every time. So finally, this is the last part of the abstract. The authors sum up the main takeaways of their paper in one sentence. It's like a mini conclusion of their research. Are there any questions so far?
Nope, perfectly clear. Thank you.
Yeah, nice. Next, we have the introduction. So this section is meant to bring the reader up to speed about what is already known in the field and to explain the rationale of the project. In other words, it answers the question, why did we take the time to test this scientific hypothesis? It also prepares the reader for the structure of the paper. I'll show you what I mean. So if we remember the abstract of the paper, we'll see that this introduction gives us background information about the different topics that came up in the abstract. So in this part of the introduction, the authors talk about CBD and which receptors it binds to. In this part of the introduction, they talk about serotonin and its implications in pain. And in this last part, the authors explain the structure of their project and which scientific questions they're attempting to answer. So the introduction is essentially the abstract minus the results, just blown up. So the authors really get into the nitty gritty and all the background information you need to understand why they're doing this project. Introductions can be quite long, but they're extremely useful at giving non-experts the background knowledge that is needed to understand the paper and why this was a project worth doing. Next, we get to the methods section. This is usually the most difficult part of the paper to understand because it is the section meant to describe every tiny detail about how the experiment was performed. It should be so detailed that anyone should be able to replicate the experiment without any ambiguity just by reading the section. It is separated into subsections for every aspect of the author's project. So here are a couple of sections you might find, and it's extremely common to find multiple of these in a paper. So you see drugs, animals, cell cultures, viruses, surgery, more, and there's infinite many different subsections you can find in a paper. So the authors must explain everything. So this is the animal section of their methods. And so here they have to explain exactly which strain of animal they use, their weights, the conditions in which the animals were housed, the fact that the experiment was approved by an ethics committee, what time the experiments were performed. As I said, someone should be able to perfectly replicate the experiment just by reading this section. So this subsection talks about the different drugs the authors use, and it includes where they got the drugs from, what it does, and the solution in which it was dissolved. Here, the authors describe the behavioral experiments they use and the exact conditions for each of them. There's a lot of other ones, but this is the last one I'm going to talk about. So there's always a section for statistical analysis in any original research article. If you're unfamiliar with experimental statistics, it might seem like gibberish, but in short, this paragraph talks about what statistical tests are used for which experiments. This is important because even if someone was able to replicate your experiments and collect the exact same results, they must be able to get the same statistical significance as you. And this section explains how to do that. I've only showed you a couple of paragraphs of this paper's method section, but it's way longer. And a lot of these details are not very useful to a reader that simply wants to understand the results of this paper. For example, if all I cared about was the effect of CBD on pain, it's not very useful for me to know that the temperature of the room in which the rats were housed was exactly 20 plus minus 2 degrees Celsius. So when you get to the method section of a paper, you should keep in mind that understanding every intricacy might be beyond the scope of what is necessary for you to understand. Now we've reached the results section. Here, the authors describe the main findings of their experiments. The results are usually split into subsections, which are each a piece of the puzzle, and describe the results from a specific part of the project. They are often presented in figures, which are graphs, charts, or any graphical aid that lets the authors visualize their results. The results are always presented with the statistical analysis used on the data. So the stats are usually put in brackets in the middle of the sentence and often seem awkward when you're reading it, but they're extremely important. Here's an example. So if, I don't know, 30% of kids that ate, we'll go back to the chocolate and obesity example. If 30% of kids that ate one chocolate bar a day became obese, but we know that 27% of kids that ate no chocolate still became obese. Can we confidently say that eating a chocolate bar a day necessarily increases the onset of obesity? Or were those findings, or were those kids that became obese going to become obese regardless? Performing statistical analysis lets us answer scientific questions in an objective way. So I won't, do a statistical analysis on this chocolate example. But the point is, experimental statistics essentially give us the probability that our results happen by chance. So the gold standard with which a researcher can say that their results are genuine is this thing called p is less than 0.05. And you'll see this come up a lot in the paper. In other words, the statistics indicate that there is less than a 5% probability, p, that the results they got happen by chance. The reason I want to explain this is because when we see some of the results in the next slide, we can look for this p is less than 0.05 and say, okay, chances are these results are legit and they're not by chance. This section of the results talks about how CBD lowers the activity of serotonin neurons. If we look at some of the more ambiguous terms, let's try to understand them with what we now know about statistics. So we see two-way repeated measures ANOVA. All this is simply the statistical test they used. We see the famous p less than 0.05. Well, over here, we also see p is less than 0.001. I don't know if anyone wants to venture on, what that may mean based on what we know on that p less than 0.05 means less than 5% chance. But essentially, with the p less than 0.001 means that there's less than a 0.1% chance that the results that the authors found happen by chance. So we know that the results are very strong and reliable. Another thing we often see is n. So here we often see n equals 9. And what that means is that the authors used 9 data points in this statistical analysis. So this 9 data points could be 9 rats or 9 neurons or 9 anything, 9 kids who ate chocolate. There are some things in this slide and that you'll find in other papers' results sections, but understanding what we've just gone over allows you to understand what is fundamental to the paper. Here we see another results section, but this time it's about the effects of CBD on pain. And again, we see some of the common things. Here we see p less than 0.001, the name of different statistics they use, n equals 6. So these things keep repeating themselves. So not just in this paper, any paper you'll find more or less the same thing. So this is a figure that's meant to visualize the results. In the results, the authors will often reference their figures by putting in brackets, something like figure 4a. And if you were to go to figure 4, you would look at what is beside the letter A to understand what the authors are trying to get you to see. So for example, if in this paper, the authors in the middle of their results section, they'd say, the scheme and timeline of our experiments are found in figure 4a. Okay, so this is figure 4. We go to A and we see, okay, here's a graphical representation of what the timeline is. So we see at each day what they do when they inject CBD or other drugs. And all the other figures are just representations of the data. With every figure, there's a legend, which essentially explains each part of that figure. For example, in this legend, we see the letters A through J in brackets. They're scattered all over the place. So here's A, here's B, etc. And the explanation that follows each letter is meant to describe the corresponding graphic in the figure. So figures should usually be self-evident, but reading the legend can be useful if you can't understand every aspect of the figure. At last, we get to the discussion. So this section lets the authors interpret their results and explain how they fit in the context of what is already known. It is usually a continuation of the introduction, but now incorporating the results of the paper. Authors often discuss how future experimenters should consider the results when designing a related project. So what often happens is, if, for example, I was interested in CBD and pain, and I read this paper, I would look at the results, and I would try to take a hint at, okay, these authors think that these future experiments are worth pursuing, so maybe that's something I could do. Now I'd like to give you some tips on reading a paper that, for example, I don't know, you're assigned in one of your classes and it's difficult to understand. So now I'm going to stop explaining different parts of the paper. I just did that. Does anyone have any questions, or they want me to go over anything specifically?
We have a couple questions.
Mm-hmm.
So I think this is just about to be answered in this section, but there was a question about how you might approach reading a research article, because we mentioned talking about the abstract, then the intro and methodology and so on. So would you approach it that way, or maybe start with the methodologies? That was one question.
Yeah, it's a great question. I'm about to get into it right now on this slide. So
perfect. So just to ask the other question, someone asked whether that statistical analysis might be used for qualitative data, because it makes sense with quantitative data, as the example showed, but would it also be applicable if it's like qualitative, and are there proofs of reliability with that?
Yeah, absolutely. So any study that's ever been done, for it to be published, needs statistical analysis, because without that, you don't know, are these results that we find, is it a trend, or is it by chance, or are they legit? So we need statistical analysis. With qualitative data, you need something quantitative to use statistical tests on. So what often happens is they'll convert this qualitative data into something quantitative to be able to put that in. So I can't think of an example at the top of my head. If you're thinking about something specifically, whoever asked the question, you can bring that up. But in general, that's what happens is there'll be something qualitative, but they'll just translate that into some kind of a number, a scale, which they'll be able to put into statistics.
Perfect. Thanks so much.
No problem. So to answer your other question, here are tips on reading a research paper. So the first thing is to focus on the title and keep it in mind as you read the paper. The title is meant to summarize the main finding of the project. So it's very useful to keep that in mind when you're reading a section you don't understand. You can ask yourself, okay, how does what I'm reading relate to the title? Next is the abstract. If you understand the abstract, then you understand the design of the paper and the results. So it's really important to keep rereading the abstract until you are confident that you understand the flow of the paper and how each result will tie into the story that the paper is telling. So often when I read a paper, especially in a field that I'm not experienced in, I'll read the abstract many, many times until I get a good grasp of, okay, I'm confident, I understand the general scheme of the paper, I'm ready to start reading the actual paper now. When you get to the results section, it's very useful to keep looking at the figures as they're referenced. So doing so allows you to visualize the findings and will help make the results more clear. So when you're reading the results, like in some of the slides I showed, some of the things can seem a bit abstract. So when you're seeing, okay, P is less than 0.05, they found this and this, it might seem like a lot of information. But when you see a graph or a chart, the point of making these graphs and charts is to make things more clear. So looking at both simultaneously really allows the results to come to life and just be more easily understood. So next is the discussion section. It's important to be critical when you read a paper. So after you understand the results, read the discussion carefully. So does the interpretation of the authors correspond to their results? Another thing to pay attention for is whether or not the authors discuss the limitations of their paper. So for example, if a study only uses male animals, something that's often the case, and there's a lot of politics involved in this, was this done on purpose, or were female, I don't know, mice or rats, just not considered? And similarly, do the authors generalize the results when they shouldn't have? So males and females, specifically looking at, I don't know, mice, for example, they have fundamental hormonal differences that influence their brain and behavior. So perhaps a brain region in male and female mice might respond differently to the same drug. The point I'm trying to get across is that the limitations of one's research, there's always limitations, they always exist, and it's important to acknowledge them even if the authors don't. And this you get better at when you're actively trying to do it. So if you're just reading the paper, it's easy to not think of the limitations of the paper, but when you're actively trying to look for, okay, the authors could have done this differently, they didn't summarize this well, you become better at realizing what the results tell us and what they don't tell us. Lastly, reread the title and the abstract. So do these accurately summarize the paper you just read and understood? So the title is meant to put a whole research article in one sentence. So the research article could be, there's some that could be, I don't know, 20, 30, 40 pages. Putting that in one sentence for the title requires a lot of concising and condensement, and and it's very easy to lose a lot of information when you do that. So when you read the title, say, okay, is this a good representation of what the actual paper tells us? So these are tips on reading a specific paper, for example, that you're assigned. So here I have a few tips on researching a topic. So this is more useful if, I don't know, you're giving a presentation on a general topic, or you just joined a lab and you want to get up to speed about the research they do in the field in general. So first, and the most important thing is know what you're looking for. It's way easier to search for a paper with specific filters than to hope you stumble across one. So for example, if I was looking for the paper we discussed today, I wouldn't just search CBD. I would look instead for papers that talk about CBD, serotonin, pain. If I know the authors, I would include the authors in my search. Next, try finding an appropriate literature review. This is always where I start when I'm trying to come up to speed in a specific field. Because finding a review that's related to your research question allows you to get a summary of what is already known about it. And it will give you links via the references to specific papers that might interest you. So when I first joined my lab, for example, I know they did a lot of research on LSD. So the first thing I did is I found a literature review on LSD. And then what I did is I looked at the references, or the ones that were mentioned that I found interesting, and I just read those papers. And you get in a cycle of, okay, I read a paper and I read the papers that this references. And that way you won't spend too much time, trying to find papers and saying, okay, is this useful to me or not? Then I read the introduction and discussion. Okay, so when you finally find a research paper that matches your search, so for example, I had a paper and I used the reference to find another paper. This is how I like to quickly read it to extract the gist of the paper. So I'll read the abstract to understand the flow of the project. I'll then look at the results, which interest me, and the corresponding methods in order to see if the results were obtained in a way that fits my search. What I mean by that is if you're trying to do a review, if you're trying to understand a specific method, a specific behavioral test, for example, and I'm trying to find different papers that use this behavioral test, what I'll do is I'll look at the methods and read the way the authors explain the test I'm looking for. And if it's exactly what I need, I'll take that into consideration. I'll bookmark the paper as something useful. I then read the introduction and discussion, and I take note of the references that might be useful to me, like I just mentioned. While I do all this, I take a lot of notes. When you read a paper, it's easy to fall into the trap of thinking, oh, I don't need to write this down, I'll remember it tomorrow. And then a week later, when you need access to the paper you have, you need to spend an extra 10 minutes trying to find it again because you didn't take any notes. So taking notes is extremely important. Does anyone have any questions on these tips or want me to go more into depth on something?
Well, I was just wondering, did you use a lot of these tips more so for research that you had to do in school, or did they develop more when you had to do research related to your lab?
So when I was in my undergrad, I wish there was a seminar like this, because you weren't really taught how to read a scientific paper. So you're pretty much left in the dark and you just have to read it, and it might be struggling. You just keep reading it and reading it until you try to understand it. But all these tips I've developed just through practice. So I've read, many papers trying to understand different things, trying to, be more informed about things of my field now. And I've, assembled these tips just by reading a lot of papers. Yeah, so that's all I have. Are there any questions about, what I presented today or about research in general?
Now, if anybody wants to add to the chat, you definitely can feel free to. Otherwise, you can unmute yourself and ask as well. In the meantime, I was wondering if you might be able to give us a little more information about the findings of your research that you mentioned in the beginning, because I thought that was really interesting, and I'm sure a lot of people would be interested to see how that might, like how a research project can go from one stage and then now you're using the same thing to go into your master's. I think that's really cool. Do you mind giving us some more information about that?
Yeah, absolutely. So when I first joined my lab, how it worked is I just email blasted a bunch of different labs that I found interesting, and I hoped that I would get interviewed to one of them. So I was lucky enough to be interviewed here, and over here, this lab does a lot of research on hallucinogenics and drugs like CBD and THC. And I found that really interesting. So when I came, I was part of a project of my supervisor, Dr. Danilo De Gregorio, and his research focused on LSD and trying to understand the mechanisms with which it works. So pretty much what I did when I first got here was just a behavioral test using LSD in mice. So first I just got good at handling mice. I did a whole bunch of workshops and I got the ethical approval to handle mice and administer substances to them. And there's a lot of just repetitive, tedious work towards the beginning. And then the more familiar I got, the more I was able to actually firsthand perform experiments. And so for 2 years, I've been doing research here. And essentially what I found during my undergraduate research was that LSD does in fact have pro-social effects, meaning that mice in general like to interact with other mice, as do humans. And we found that LSD increases their preference to interact with other mice. And the reason this is important, it might seem like some random thing, who cares? But the reason it's important is because there's a lot of mental illnesses where antisocial phenotype is very common in them. So like, I don't know, things like depression, anxiety, autism, all these things often lead to people self-isolating. And so the grand scheme of this work is hopefully one day we can implement what we found into humans.
Wow, that's incredible. So exciting. You'll have to let us know how the new research goes.
I will.
We have some more questions, but could we turn it back to stop sharing the screen?
Yeah, for sure.
Perfect. Perfect. So some of the other questions included, how do you take notes? Do you usually make a separate document and paraphrase, or do you highlight important parts by hand on the computer? What are some of your preferences usually?
So the way I like to read papers is by printing them. I think it's the easiest way, actually having it in front of you, a hard copy, and like that you're able to highlight what you need. So this is the way I personally like to do it, but you could do the same thing online with PDFs. There's ways to highlight, different text. The way I take notes though is after I've highlighted, I'll reread a paper and specifically focus on what I've highlighted in the first place. And then what I'll do is I'll open up a Word document and essentially write the results that interest me. So for example, this last paper, if I'm trying to do, a literature review on CBD, what I'll do is if, for example, I read this paper, I would say, okay, CBD was found to do this for pain, this for pain, this for pain, and I'll cite the paper. So I'll put a little superscript or like a 1, and at the bottom, I'll keep a list of my references. And the reason this is important is because, I won't just be using one paper. I'll be reading a lot of papers and, incorporating many different results. And when it's time to actually cite these things in my own paper or just, I want to reread some of them, if you just have the different results, it's easy to forget, oh, where did I get that from? So, referencing them and giving them superscripts allows you to go back easily and find them. So that's usually what I do. I like putting notes in a separate Word document. Just like that, they're very, condensed and just what I need.
That's amazing. Yeah, I definitely feel like that's good advice. From my own little exploring of research, I always get lost in all the different research that I'm doing. So keeping notes, I can also vouch for that being a really good technique. We have some more questions. Oh, well, first of all, a comment saying that this was really interesting for a good way of analyzing, so like reading analysis method, and it's actually making people feel more comfortable reading scientific papers in the future. So we just really thank you so much. More questions about, so now you're moving on to your research. People are asking if you could talk a little more about what you're doing right now in terms of your research and like where you hope it to go.
Yeah, absolutely. So
kind of
I think it's
mentioned a little bit.
the details of it because a lot of it is unpublished work. But essentially, we're trying to use the information we found into genetic models of actual diseases in mice. So, for example, you could model. There's many different genetic ways to model disease. So you could model. I don't know. You could do cancer, autism, depression. So many different things in mice, and doing so allows you to test. What you're looking at. So for me, it would be LSD in these genetic models of these diseases. So it's not necessarily 100% translatable to humans, but it gives you it gives you a lot of insight on where could the research in humans be heading in the future. So so essentially what I've been doing now is we have a genetic model of a certain disease in animals that displays a strong antisocial phenotype. And we're seeing if, okay, what we found about LSD can be applied, to this genetic model.
Very cool. Um, so even more questions. Do you have some words of wisdom for students trying to get involved in research? A lot of the students that we have reached out to have never even done research before at all. Do you have advice for a first-time interested student?
Yeah, absolutely. So it seems like a very daunting thing to, put yourself on the line and try to get research experience, especially when you don't have research experience. So that's kind of the big conundrum. It's not just, it's about work in general. Like, how do you get work? You need work to get work, but you can't get work because you don't have work. So the best advice I could give is just to realize that it's a numbers game. So send emails to as many PIs so researchers that you can, and you're essentially hoping that they're looking to recruit someone. So I'll give you an example of what happened this past summer. Unfortunately, because of COVID, a lot of research projects didn't come out. But I was applying for, there's this grant at McGill that actually I encourage everyone to apply for. And essentially they pay, forget the name of it, but it's very easy to find if you go on the McGill fundraising webpage. Essentially what they do is they'll pay you a certain amount of money to go travel somewhere in the world and perform research at another institution. So when I first heard about this opportunity, I said, oh, okay, this would be a great chance to go to one of these, schools that's so well known and has a strong reputation for neuroscience research. And like that, I could learn stuff there and perform good research there and bring that back to McGill. So what I did is I sent emails to 50 or 100 different professors. It was really, really time consuming. The best way to do this, it takes a lot of time, but I guarantee you it's worth it. It's find the PI, so find the researcher whose lab you'd like to join, read a lot of their papers. So look at them up on PubMed and read, usually they'll be the last name, they'll be the last author of the paper. And what you want to do is read the papers, find one that really interests you and that you really understand, and send them an email and say, hey, look, I would say, I'm Tommy, I'm an undergraduate research student at McGill, I'm studying neuroscience. I really admire the research you've done with this and this. I think this is a great way, I think this might be very impactful in the field of whatever. You essentially want to demonstrate to them that you took the time to understand their research and that you're committed to being involved in their project. So I did the same thing and I applied, to a ton of different ones. Out of the hundreds of universities that I emailed, I got one response from a researcher at Stanford. And we were at, it was all lined up, but unfortunately because of COVID, it didn't go through. The main point is that it's a numbers game. And the more you send, the more likely you are to actually find a position. So I encourage you to do that. Even, it doesn't have to be only with this grant I mentioned. It could be even at McGill, just trying to volunteer your time, or like I mentioned at the beginning, to do an undergraduate research class.
That's great advice. So on that note, we have more questions. If with this grant, is that an experience that you would be able to have paid research work for, or is that volunteer and then the grant goes towards the research?
No, they strictly
Like,
pay.
do you get paid?
Yeah, they don't pay for the research. So they pay you to be able to afford to live in wherever this foreign university is. So it's quite a large sum of money that they do give you. And the logic is they'll give you this money and it's meant to cover rent and food and, travel. No, the research that you actually perform is always funded by the actual researcher. So it's not something, you need to worry about.
Okay, perfect. And are there other opportunities that you know of in terms of a research position that is paid work? Because people are saying that usually research opportunities are like, you might be able to find them, but they're much more frequent as volunteer positions rather than paid positions.
Yeah, so I find that usually what happens is you'll find a lab, let's say you do find a lab that wants to take you on, especially during the school year. The reason it's a little tricky for them is because they need to structure the way they pay you it's very structured the way this has to happen. So it can't just be like, okay, like come in and I'll pay you however many hours you come. From what I understand, it's, you know, very structured. So what happened with me when I first joined my lab here is I got accepted in January. I said, okay, like they told me you can come in and, you know, just learn the ropes a bit. So from January until the summer, I didn't get paid. I just volunteered my time to learn the techniques and show them that, you know, I'm committed to this research and I'm really investing my time in this. And then that summer, there are different ways with which your researchers can pay you. There's, I think the official title is summer research student or something along those lines, and they could in fact pay you. I think all this information is not too hard to find on the McGill website. Yeah, but it's, you know, you might encounter a lab that doesn't necessarily have this kind of funding. They might not be able to pay you, but my personal opinion is even if they don't, I guarantee you it's really, really worth it, even if you don't get paid.
Definitely. And we have an experienced person attending here saying that also other summer paid positions include NSERC, SURA, and SURE grants. So please, if anyone's interested, you can message us or Tommy or keep in touch and look yourself as well for different opportunities. There are paid positions, but as Tommy mentioned, it's very valuable to even do the volunteer positions. Last question, you mentioned these the school courses, the courses that are related to research. Can you explain a little bit more about what those are, how the profs are, what it really kind of is like going to that? Is it an elective? Does it take a lot of time?
So, okay, so classes, I could not recommend them more. So the one that I took is called MSWI 420. So this is it's specific to neuroscience, but I think every single science program has one of these. So I know, for example, there's absolutely one in psychology, there's one in anatomy, there's one in physiology. I'm pretty sure they all have one. And the point of this is to expose you to independent research. So the way these classes usually take place is you have to independently find a PI that wants to take you on. So that's more of the hard part. It's not really, it's not like there's a limit of how many spots are open in the class. It's more, can you find a researcher that wants to take you on? So that's when, you know, it's important to email a lot of people, play the numbers game. Once you get accepted from a lab, there's a little paperwork that needs to go on. So you would talk to your PI about, okay, what your project is, what roles would you do, what techniques would you learn to do? And actually registering for the class once you have a PI is very easy. So now the way the class works is there's no test or anything. Pretty much your whole grade comes from your PI. So there's no teacher involved. And, you know, they grade you on your lab performance. So again, this must vary class to class. The way mine was is that 50% of my grade is just lab performance. So if my PI liked the way that I, you know, conducted myself at the lab, I was reliable, I didn't come in late, for example, I would get a good grade for that. And 50% is I had to write a manuscript on my paper. So all these research papers, you know, articles that I just described today, how to read, that's essentially the end goal of doing research. Like you want to do research and you want to get it published, so it's out there in the literature and you've contributed to the scientific community. So what these classes allow you to do by writing a manuscript is essentially an unpublished research article. So you still write it, you know, it's very structured. Like mine, for example, included all the sections I spoke about today. It's quite long. It's a lot of work, but it's really, really worth it because it really, you know, you get your hands dirty and you really learn how to you know, complete a scientific project from beginning to end. And that too is graded by your PI.
Wow, very cool. And the PI has to be from McGill as well?
I believe so. There might be some leniency. I know from my program, I think it had to be from McGill, but again, don't take my word for it, but you might be able to get away with other universities. But it should be outlined when you look up the class.
Awesome. Okay, that sounds awesome. I'm definitely going to be looking into that, and I hope to see people attending this event with me because that's really cool. Yeah, so if ever anyone has any other questions for the next minute or two, you can still write in the chat. Otherwise, as I mentioned before, you can definitely message us on our Facebook page or through our email and visit our website, check out our ebook, and we will be having monthly workshops. And hopefully we will see Tommy again at a future workshop because that was just amazing. Thank you so much for coming.
Thanks for having me.
Perfect.
Thank you, Tommy. That was awesome.
Perfect. I'll stop the recording.